Hi everyone, welcome to our channel. In this video, we are talking about the pure cultures and the isolation methods. Firstly, pure cultures. As we know that microorganisms occur in nature in extremely large population, and they are present in very small amount, and their size is very small, so we cannot see them with our naked eye. In order to study the characteristics of a particular species, we firstly have to separate specific one organism to separate the specific organism from all other organisms. For this, laboratory procedures have been developed that make it possible to isolate microorganisms representing each species and grow each of the species separately. For example, we have a Petri plate and we have several organisms that are of different species, like one or three, and we want to study the specific organism. Firstly, we have to isolate this organism from all these organisms. The growth of a mass of a cell of the same species in a laboratory vessel or a test tube or any other equipment or instrument is called pure culture. Now, the history of pure culture. Pure culture of bacteria was first described by Joseph Lister. He used serial dilution in liquid media. With a specific constructed syringe, he diluted a fluid. He used probably a milk sample containing mixture of bacteria until a single organism was delivered into a container of sterile milk. After incubation, bacteria in this container were of a single kind, identical to the parent cell. Lister named that organism as the Bacterium lactis. Now, the isolation of pure cultures. Now, for example, we have a Petri plate and we have several different types of organisms. One species identifies as the blue color, while the other species identifies as the red color. And if we want to separate these two bacteria or the cultures, we have to take a looful of red culture, and streak it on a separate plate. Streak it on a separate plate like this, or any other technique. Or a looful culture of blue culture, and take a looful of blue culture and streak it on another plate, like this, or any other streaking methods. Then incubate these two plates for 24 hours or 37 degrees Celsius. We have find the pure cultures like a blue culture and like a red culture. Now, the purpose of pure culture. As developed by Robert Koch, pure cultures allow the pure isolation of a specific microbe, which is vital in understanding how an individual microbe may contribute to a specific disease. For example, through pure cultures, we can identify the specific organisms like bacteria, how they can cause a specific disease, and through this we can identify their prevention, their measures, and their treatment as well. Thank you. That's all from my side. Thank you for your watching.